In this video, I want to talk about hemoglobin, right? Again, abbreviated as HB. I want to talk about the Bohr effect, and I want to talk about fetal hemoglobin, which is abbreviated as HbF. Okay, and how all these sort of play in together in the grand scheme of things. Okay, so in the last video I mentioned allosteric regulation when I talked about hemoglobin, right? I mentioned this idea of allosteric regulation and cooperativity specifically. Now, allosteric regulation, again, is just as a brief sort of definition, is the whole idea that the binding at one subunit of a multi-subunit protein can have an effect at the other subunits as far as affinity of that for that ligand goes. So there is the case, of course, that when one oxygen binds a particular subunit, the other subunits have an increased affinity for oxygen. Now, there are other things that can bind hemoglobin that will actually reduce the affinity of hemoglobin for oxygen. So there are three things that I want to mention. So there is H+, right, which is just protons. There's carbon dioxide, and there is this thing called 2,3-bis-phosphoglycerate, otherwise known as 2,3-BPG. Okay, now all of these things here, all of them, they all reduce hemoglobin's affinity for oxygen. Now how do they do that? Okay, so hemoglobin, this is something I didn't mention in the other video. Hemoglobin has two states that can be stabilized. Okay, there are two states. There is the R state, or the relaxed state. Okay, so hemoglobin can be relaxed, or it can be in the T state, which is the tight or taut state. Or taut state. Okay, what do you need to know about these two states? Well, essentially, what you need to know is that the R state has a high affinity for oxygen. Okay, it binds oxygen tightly. The T state has a low affinity for oxygen. Okay, so it actually releases oxygen. So this binds O2 tightly, and this one releases oxygen. Okay, so now if all three of these things here, right, if the protons, carbon dioxide, and 2,3-BPG, if they're all reducing hemoglobin's affinity for oxygen, what they're doing is that they are stabilizing the T state. Okay, so all three of these things, right, they stabilize the T state of hemoglobin. Okay, so let's talk about sort of how this works. Okay, numbering each one at a time. Okay, so let's do proton concentration. So proton concentration, essentially what happens is that if we have hemoglobin bound to oxygen, right, which we kind of denote like this, HbO2, if we add H pluses, right, protons, we shift the equilibrium to end up having this, which what we get is if we add enough protons, what ends up happening is that the protons end up binding the hemoglobin, and then the hemoglobin releases the oxygens. Okay, so just Le Chatelier's principle back to general chemistry is that if you add enough of a particular reagent to one side of the reaction, you will shift the reaction to the other side. So if we add enough protons here, we will shift the reaction this way so as to stabilize the T state of hemoglobin, causing it to release oxygen. Okay, so essentially what we're trying to talk about here is that, excuse me, is that as pH, actually before I mention pH, as H plus concentration increases, right, we favor the T state. We have more hemoglobin in the T state, right? So what that means is that more oxygen is released. Okay. Now, pH increases are the same as, or excuse me, I think I might have just, I don't know what I just said, but let me just say it again. H plus concentration increases is the same thing, right? This is the same thing as a a pH decrease. Okay, so a pH decrease is the same thing as an H plus increase, right? So decreased pH, increased H plus concentration causes more of the hemoglobin to exist in the T state, and more oxygen is released. So when does this sort of this concept come into play? Excuse me. Actively metabolizing muscles, for instance, actively metabolizing muscles have lactic acid buildup. Now, what is that? You might have heard of, when you exercise, you get like soreness in your muscles. That soreness is due to lactic acid buildup. The details behind that are not too imperative right now. The whole idea is that if you have lactic acid buildup, then acids give up their protons, right? This will increase H plus concentration, decrease the pH, and we have more hemoglobin in the T state, so we end up with oxygen being released from hemoglobin. Okay. The second thing, carbon dioxide. How does that work? Okay, what happens there? Well, if you recall, in the past we mentioned this sort of reaction. Okay, this whole idea that we can have carbon dioxide plus water yielding carbonic acid, which will dissociate into H plus and bicarbonate ion. Right? The enzyme that does this reaction, I think I mentioned it before in a previous video, is called carbonic anhydrase. In any case, what we talked about before is that if we increase the amount of carbon dioxide, the reaction will shift so as to create more H plus ions, right? So more carbon dioxide equals more H plus, right? Which is just the same thing as a decreased pH. Again, same idea. More hemoglobin is in the T state, right? So more oxygen is released. So it's a sort of the same idea as above, as mentioned above. Okay. Another thing is that actively metabolizing cells create CO2 via cellular respiration. Right, this is something we'll mention later. But this, if you recall, we breathe in oxygen, right, and then when our cells use up oxygen, and then we breathe out carbon dioxide. Before we can breathe out that carbon dioxide exists in our tissues. So cells that are actively metabolizing oxygen are creating a bunch of CO2, right? So if we're actively metabolizing cells, we're going to have a lot of CO2. Then that's going to lead to a lot of H pluses, which is going to cause the oxygen to be released to those cells, and that makes perfect sense too, right? If our cells are actively metabolizing, they're doing a lot of work. They need a lot of oxygen, right? So we want to make sure that when the hemoglobin gets to our tissues, that there is a high concentration of CO2 that causes the oxygen to be released to our tissues so our tissues can use them. Okay, so hopefully that makes sense there. Okay, now let me sort of break that down in a little chart before I actually get to discussing 2,3-BPG. So essentially what we got here is we've got lungs, right? Our lungs, and we've got actively metabolizing tissues. Right? So in our lungs, we have a high pH, right? High pH relative to the actively metabolizing tissues. Right here we have lower pHs, right? Due to the production of H pluses, right? In the two ways listed above. Okay, so now this makes sense. Why does this make sense? Well, because in our lungs, right, we want hemoglobin to bind oxygen, right? So we want there to be a high pH. We don't want a low pH there. If there's a low pH there, that means hemoglobin can't bind oxygen. If there's a high pH, then hemoglobin binds the oxygen, right? Here, the low pH causes hemoglobin to releases the oxygen. Okay, which is what we want. In the lungs, we breathe in oxygen. We want the hemoglobin to bind the oxygen. Then we want that hemoglobin to take that oxygen to our cells, right, or our tissue, actively metabolizing tissues, and we want that O2 to be released to our tissues. Okay, so in the lungs, where there's a high pH, right, hemoglobin releases the H pluses, right, whereas here in the actively metabolizing tissues, hemoglobin binds those protons. Okay, hopefully that sort of wraps things up and makes sense overall in terms of our physiology. Okay, the third thing is of course 2,3-BPG, right? 2,3-BPG is the structure here. Let me actually draw this out for you. So 2,3-BPG looks like this. It's got a carboxyl group and then a CHO phosphate group here with these negative charges, and then it also has a CH2 and then another phosphate group. Okay, this is 2,3-BPG. Okay, so what do we notice about 2,3-BPG? Well, it's got a lot of negative charges. Okay, so this is important. Okay, the reason why is because 2,3-BPG, what it does is it binds or interacts with positively charged things. Okay, right? What have we been talking about that can be positively charged in hemoglobin? Well, amino acids, right? Basic amino acids. We mentioned histidine, right, being a side chain in these heme groups, right? So what ends up happening is that 2,3-BPG can bind the positively charged histidine, right, or even lysines, right, that are other basic amino acids that are in the hemoglobin. Okay, because these negative charges are attracted to these positive charges, because opposite charges attract, right? So 2,3-BPG, right, will bind, and we said up earlier that it inhibits the hemoglobin from functioning. So again, 2,3-BPG, right, when it does this, it alters the conformation of hemoglobin. Oops, conformation. Sorry about that. Conformation of hemoglobin, right? Specifically, it stabilizes the T state, right? Again, causing O2 release. Okay, so if there's a lot of 2,3-BPG around, the O2 is released. If there isn't very much around, O2 is bound. And it's actually, a very important allosteric regulator of hemoglobin function. Okay. Now I also mentioned, I mentioned up here that I wanted to talk about the Bohr effect. this whole concept here is like everything that I've mentioned from here, sort of through what I just finished talking about, is kind of the Bohr effect, the whole idea behind these, the allosteric regulation of hemoglobin. Okay, now what I want to talk about is fetal hemoglobin and how that comes into play. Okay, so let me scroll down just a little bit more. So, what are the things that we need to know about fetal hemoglobin? Okay, let's bring in a new color. I don't know. Let's do baby blue since we're talking about babies. So, fetal hemoglobin, HbF is how it's sort of abbreviated. Fetal hemoglobin. Fetal hemoglobin, I'm just going to straight up say it, that it has a higher affinity for oxygen than maternal hemoglobin. Has a higher affinity for oxygen than maternal hemoglobin, or just hemoglobin. Right, this is just Hb. Okay. In case it's not clear, adult hemoglobin is just maternal hemoglobin. Okay. Why is this? The reason for this is because fetal hemoglobin has a lower affinity for 2,3-BPG. Why? Why is that the case? Okay, that's what we're going to talk about in just a second. Okay. And so I do want to note, though, before I start to explain things, is that its function is to deliver oxygen from the mother to the fetus, right, to a pregnant mother's fetus. Okay, so now I just mentioned that it has a higher affinity for oxygen than maternal hemoglobin because the fetal hemoglobin has a lower affinity for 2,3-BPG. So what is going on there? Okay, so let's mention this. Let's mention more details about fetal hemoglobin. Fetal hemoglobin, excuse me, its structure. It also has 4 polypeptide chains. Okay. But instead of two alpha groups and 2 beta groups, what it has is 2 alpha groups and 2 gamma groups, 2 gamma subunits, 2 alpha subunits, 2 gamma subunits. Okay, and that this is what makes it different than adult hemoglobin. Okay, so it binds this 2,3-BPG more weakly. Okay, because it binds this more weakly, how does that sort of work, right? What is the difference there? We said that 2,3-BPG is an inhibitor of hemoglobin function, right? So if this essentially means that if you're binding less 2,3-BPG, right, that means you're inhibited less, right? So 2,3-BPG doesn't have as drastic an effect on fetal hemoglobin as it does on maternal hemoglobin. So it is less inhibited than maternal hemoglobin. Okay, which if it's less inhibited, that means that it can bind to oxygen better. Quite simply, right? So why is this? Why can it do that? Well, this is the sort of the boom here. Okay, these gamma subunits, right? What's going on there is that a serine residue, right, which is a polar but neutral, right, a polar neutral amino acid, is substituted, right, in place of a histidine, which is a polar but positively charged amino acid, right? So what's going on here? What's the deal? It's quite simply is that this negative charge, right? The negative charge of 2,3-BPG is more attracted to the positive charge of histidine right, than it is to the neutral the neutral serine, right? Okay, so essentially what happens is that 2,3-BPG cannot bind fetal hemoglobin as tightly as it can maternal hemoglobin, so fetal hemoglobin is not inhibited in the way that maternal hemoglobin is, so it has a higher affinity. And just to show you how that would look on the O2 saturation curve, if we have maternal hemoglobin, it kind of looks like this. Actually, last time I drew it in blue, I might as well draw it in blue this time. So maternal hemoglobin looks a little bit like this, right? If this is maternal hemoglobin, right? And of course, these axes are still labeled as the pressure of oxygen and the percent saturation, right? Fetal hemoglobin, of course in the baby blue, would be shifted to the left, right, of maternal hemoglobin because it has a higher affinity, right? It doesn't need as high a pressure of oxygen to become more saturated. So for instance here, again, just a sort of quick example, is that if we have, let's say this here is, I don't know, like 10, let's just say it's 20 torrs of pressure, right? If this is 50%, this is like 100% up here. These are just numbers I'm making up at this point. But at 20 torrs, hemoglobin, right, is less saturated, right, than fetal hemoglobin, right? Fetal hemoglobin might be at like 25% maybe, and whereas hemoglobin will only be, I don't know, maybe around 10%. The whole idea is that fetal hemoglobin can be at a higher level of saturation at a lower concentration of oxygen, which means that it has a higher affinity. I hope that video was helpful. See you next time.